Okay, good afternoon, everybody. How are you all? Good? Beautiful day today, right? So, I want to start off with a little announcement. I asked at the last but one presentation for suggestions of speakers for the spring and fall next year. I had one email from you guys, so I sent an email out. I hadn't had a reply from that person yet, but if you've got ideas of people you'd really like to have brought to campus for the spring and fall series, please email me at mlee@calstate.mll.edu. Okay, so I want to, today we're going to have a very interesting presentation from Nate Jew from the CSU Monterey Bay, and he's going to be talking to us about using de novo assembled genomes to inductively explore the biology and evolution of marine invertebrates. Give you a little bit of an introduction to Nate. He's being sponsored today, the guest of the Invertebrate Zoology Lab, but I'm going to do the presentation on behalf of them. And so Nate did his undergrad at Colby College in Maine, and after a brief foray into teaching high school English, because he also did a minor in creative writing while he was there, he ran away to warmer weather to be a marine ecologist and work for the state of Florida on marine reserves in the Florida Keys for a few years, and then his interest in larval dispersal got him looking to get a graduate degree related to marine species population genetics, and so he ended up at Florida State University, where he was converted to more of an evolutionary biologist after joining Joe Travis's lab and working with Chris Koenig and Felicia Coleman on the population genetics and ecology of gag groupers in the Gulf of Mexico. Been to some nice places, huh? During the end of his time there, he became interested in research questions related to how genotypes confer phenotypes, and ended up as a postdoc at the University of Connecticut in the genetics and genomics lab of Michael and Rachel O'Neill. He was working there on a project related to using the evolution of placentation in fish as a means to generally examine questions on the evolution of complexity. During his time there, he began his final transition from a population geneticist to an evolutionary and functional genomicist. Now he works on evolutionary and functional genomic projects from everything from horseshoe crabs to humans, but is still particularly focused on fishes with research questions that are still broadly related to how genetics or now genomic information informs us on phenotypic outcomes in an evolutionary context. So I think we're in for a really good dose of science today. He's going to tell us about his work and also a little bit about what he's doing now and teaching down at Monterey Bay, because it may be of interest to some of you looking for classes. So at no further ado, let's give Nate a warm welcome and fond attention.
Hi. Thanks for having me. Glad to be down here. As Michael was saying, I work generally these days in genomics and bioinformatics. It's a point of emphasis for me. I'm going to be talking today about what are basically side projects for me. I sort of do these out of just personal interest. People come and ask me if I'm interested in helping them out with something, and we're like, yeah, sure, let's work something out. So I've been working on these genome papers, but my sort of the stuff that I'll be doing research in my lab, I'm pretty new over at CSUMB, so I'm still getting my feet firmly set in the ground. Will be related to some of the things that Michael was talking about earlier, looking at the evolution of complexity. One other thing I wanted to mention was at CSUMB, one of the things I'm doing is I'm integrating a whole bunch of new classes related to sort of computational biology, bioinformatics, things like that. Some of the graduate students there have asked me to try to run a grad session at the same time, like double list one of my 400 level courses, which is bioinformatics, as a grad course. So if you were interested in that kind of class, you can email me and I can tell you a little bit about it and whether that be appropriate, because I think I'll be running a 595 this spring in association with that class. So today I'm going to talk about using de novo assembled genomes to inductively explore the biology of species. And so when we think about science, right, there's basically two ways that we sort of do science, right? There's inductive reasoning and there's deductive reasoning. And for a long time, inductive reasoning was kind of frowned upon in biology. A lot of that had to do with sort of going back in history and some kind of physics envy that we were in, where the physics people, said, well, if it's not physics, it's stamp collecting, right? And so we sort of, as biologists, we decided we had to get into a really rigorous deductive framework in order to be taken seriously as a science. And what's happened is with the advent of genomics, it's kind of flipped the process a little bit on us. And suddenly inductive reasoning ends up having a lot more power. So if we think about how we do science, like what the process looks like, I would have read this, well, it's not going to be replaced. But basically, we'll take some observations. That's inductive reasoning. We do observations. We go to nature. It's typical for biologists. We do this all the time, right? We observe a phenomena. We form a hypothesis. And then we try to execute some sort of experiment or study to deduce from our experiment or study and those observations about whether or not we can reject or accept our null hypothesis. This is a traditional way of doing biology and science. And what's happened is sort of with the advent of genomics is we kind of now, instead of this circle, we're sort of more in this spiral, right? Where suddenly these inductive processes, these knowledge and observational processes are advancing our science more than perhaps in the past we were just using this loop. I mean, we were always kind of in this process. I think a practicing biologist was always sort of in a little bit of a spiral. But because of genomics, we can really push this even farther. Because one of the main criticisms of this inductive reasoning process is that we never had enough information to really say that our observations were an accurate representation of everything that was going on, right? And the thing about genomics is basically we can get accurate observations of everything that's going on in the genome. So that's what makes it particularly powerful. It also presents a whole bunch of different problems, but what it's done is I am not, maybe some of you guys have been on subs. I always wanted to be on a sub. I haven't had a chance yet, someday maybe. But I kind of liken it to the same sort of process where what's opened up in terms of genomics and inductive reasoning is that we can now do these really exploratory studies in biology that, historically in genetics was a little bit harder to do because, we had to have, there was a lot of stuff that had to go into designing experiments to make sure that we knew our system well enough to be able to even do an experiment. Now, with the advent and changes, all these disruptive innovations and genome sequencing and DNA sequencing, we get to, do our own version of deep sea exploration in subs. And for those of us who love playing around with genetics data, it's pretty exciting, too. Even though we're just behind a computer, but still exciting. So why is genomics so attractive to us, right? Especially this inductive perspective. Well, one, it provides us a quote-unquote complete picture. Now, admittedly, complete, we'll keep it in quotes because nothing's ever completely complete, I suppose. But, basically we are able, by this complete picture, we're able to consider and describe all these unknown entities that we didn't even know if they exist or if they were important or present. And so it allows us to look at a depth that previously we've never been able to look at. And we get a lot of observations of unexpected phenomena because previously we would have focused our experiments on a very specific aspect of our experiment. Our experiment, whatever we're trying to manipulate, right? Whatever we're trying to observe. In this case, we're able to look at all the other correlated and possibly co-regulated mechanisms that could be going on all at the same time. And it's made it really, really interesting, also really challenging from an analytical standpoint. Like, how do we assess all these different processes going on at one time? So because we're able to do this, we end up looking at things on a much larger scale, so we can start looking at things that you can only think about when you look at large scales. For instance, networks of interactions, right? So suddenly we can look at large networks of interactions and how networks actually work in biology. We consider them to be less biased and more accurate, and hopefully will give us a clearer understanding of overall general trends that we might see in something. And it also makes it really a lot easier for us to study rare events that might be going on in biology that might be significant and important, because it's a lot easier to detect them if we're detecting everything. And the last thing that genomics lets us do, it lets us not only look at things from a genetic standpoint and from a biologist standpoint, it lets us, we no longer have to look at just things that are on a gene by gene level. We can actually look at the entire genome as an operating unit, right? Which is something that we've never done, or as a series of large operating units, And so this lets us do a lot of things too, or we can also look at, how it's structurally organized. We can look at a bunch of different types of elements where previously we had no way of really examining or understanding their contributions to biology. I think one of the ways that you can show how genotypes make the genomics can bring sort of unexpected information into the realm of scientific discussion. And one of the reasons why I like doing these little side projects and why they're interesting to me is sometimes you end up with some interesting outcomes, right? So I think if typically, traditionally in biology, if we were to say, how would we group these different organisms by sort of the closest related organisms, a lot of times we would say that, these guys right here would be, the jellies, the ctenophores are basal to the jellyfish, and yada, yada, yada. Sponges are closer to choanocytes because, choanoflagellates, because their choanocytes look almost exactly like look like these choanoflagellates, these single cellular organisms. And so what happened is that when some folks, even Steve Haddock, who's right up the road, was involved in this project, when they did some work on looking at did some genome sequencing, what they found by looking all the way across the genome and not just at a few genes, is that they actually find ctenophores coming off before sponges and in between these choanocytes. So there's a step in between, and they find unique gain of mesoderm or muscle, right? And the loss of nervous systems in some of these other taxa. So it really, this really sort of flips your perceptions on how you think, invertebrates have evolved compared to sort of a traditional approach. And there's some discussion about whether or not this is the case. There's some people who don't think it's the case and other people who do think it's the case. But this kind of investigation would not be possible without looking at it at a genome level. And so that's one of the reasons why I like doing this stuff, especially in marine systems, because I think marine systems are particularly interesting, at least to me. So, they're generally windows to the past. We have some of the oldest and most ancient species in marine systems. Of course, the incredible diversity. Just last year, Bob Reinick, just up the road, basically described a new phylum, right? That's crazy, but we're still finding phylum, right? So it's really, really diverse. There's some really old things there too. So there's a bunch of species that if we look at them today, they generally look how they looked hundreds of millions of years ago, which is pretty wild if we think about it. So by looking at their genomes, perhaps we can get windows into the past and see sort of what the ancestors of a lot of the extant species we have now look like. So really, really cool. And the other added benefit is there's a whole bunch of them that have really small genomes, which is great because those are really easy to sequence and assemble. But not all of them do, unfortunately. For instance, right now I'm working on a shark, which is like twice the size of a human, which is terrible. So if we look across sort of like the diversity of different types of organisms, one of the ones that I started getting interested in was this group right here, these urochordates. And why I got interested in these urochordates had to do with basically a friend of mine who was doing some work in the lab when I was at the University of Connecticut. She was getting all this data, and she was like, I really want to do a genome sequence. And I was like, that sounds interesting. And she's like, can you help me? And I was like, sure, why not? We'll do it. So we did it as sort of just a little side project. And the reason why this was here is these were supposed to be grayed out. I'm not sure why they weren't. But so basically, we're going to be looking at these sort of 1, 2, and 3 in terms of what the types of questions or types of topics we'll be interested in looking at in these genomes as we move forward. So this species that my friend Paula was interested in is a species called Salpithomsini. And Salpithomsini is a polar, it's an Antarctic urochordate. So it's related to a sea squirt, but it's a free-swimming organism. I don't think it has any sessile stage, as far as I know. So it's only going to be free-swimming. It's a zooplankton. And so this is a project that I did with a whole bunch of other folks at University of Connecticut. when I was there. Paolo was working in Ann Buckland's lab at the time. And this is actually a paper that we just got. Finally, it's in press. And we think we got the cover for it, too, so we're pretty excited about it. So what is salpothomsoni? As I said before, they're urochordates. Generally, why are people interested in urochordates? Well, a lot of people are interested in them because if we look at their early developmental stages, they look suspiciously and amazingly like vertebrates, right? And that's why they're urochordates, not chordates, so they're closely related. But then, of course, as they grow and undergo metamorphosis in the later stages, they look vastly different, right? So it's a major metamorphosis they go under. So that's one of the reasons we've been interested in them in a long time, because of their unique relationship to us as invertebrate, quote unquote, invertebrate species. Another reason why, and so we're looking at this group right here. Another reason why we're interested in salpothomsoni, well, this is a good example of sort of all the other genomes within this group. So these are all the urochordates or tunicates in here. These are all the species for which there are genome assemblies right now. So there's 2 species of Siona, which were some of the first genome sequences done. And the reason why is their genome is really small. Oikopleura has like the smallest genome out of any multicellular organism, I think, or one of them. It's really small. Petrillus slosheri was actually sequenced by some folks just up the road at Hopkins Marine Lab. And then there's ours, which is a new genome sequence, which is the first genome sequence for this group called the Thalassia. So it's got some phylogenetic importance, although that wasn't really an emphasis of the work that we were doing. If we think about this species specifically, why the people I was working with were interested in it, is they were interested in building a reference genome for their own work, where they're using a lot of measures of gene expression to look at the effects of climate change on the ecosystem. And the reason why is that, so this Antarctic species, which is pelagic in nature, is basically a competitor of krill. And they've started to see it starting to replace krill, especially as we get more warmer waters in this area. And so they're really interested in understanding the ecology of the species and how it might be representative, and some of the genetic differences among populations and responses to thermal stresses, things like that. And they've been doing a lot of work looking at gene expression under different conditions for this. But to do that work well, it's always nice if you have a genome sequence. So that's why they were interested in recruiting me. And it's got a somewhat complex life history. So it's got this, it has these solitary stages where it reproduces asexually, and then it'll aggregate for sexual for reproduction during that time. Then you'll find them delineating into males and females. The offspring, these embryos, will then develop internally of these females. So they have these internal fertilization and processes, and then eventually they'll bud off and form more asexually reproduced cells. They're generally found off the eastern part of Antarctica in this area, and this is specifically in this region was where we actually got this sample from. I unfortunately did not get to do the sampling. Paolo got to go. It'd be fun to go to Antarctica, I guess, but I haven't had a chance yet. And what we end up with a genome assembly when we did it, so I didn't do any of the data input, the data generation, so I can't take any of the credit or blame for that. I just had to deal with the data once I had it. But we ended up with a somewhat fragmented genome. It's not the best genome sequence that I've ever put together, right? But it's what we had, it's what we had to deal with. And it was about a 20x genome sequence coverage, and ended up being, so the genome size is about 600 megabases, and we ended up with a genome sequence that was about half that size, right? So one of the reasons we ended up with that genome size that was smaller than what we might have preferred, and so then left us at a very at what we would call a draft status, is that there was quite a bit of repetitive sequences in our DNA. So these are, DNA sequences that repeat themselves multiple times. And when you have long stretches of them, it's hard to include that into your genome assembly. Because you can imagine if you have like 10 things, all like 100 bases pair long, and you're trying to figure out like what order they go in, it's not going to be that easy to put it together. And one of the things we noticed too is there was a whole bunch of stuff that there was about 33% of the genome that we assembled that was seen as repetitive, And then 37%, almost 40% of our sequencing, it was so much repeats like AT, 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 things like that, or other very repetitive motifs, that it was basically too repetitive for us to even assemble. So it was just discarded. So it just tells you there's a lot of repetitive stuff in this genome. So it's kind of a tough invertebrate genome to assemble. And just to give you a sense, so our genome was about 6 times the size or 7 times the size of like some of these other urochordate genomes that have been assembled. And we also noticed that there was a significant section of this that was actually rDNA, or ribosome, DNA that codes for ribosomal genes, which was, will come up later. But other than that, it had sort of like your normal suite of different repetitive elements, which was kind of interesting to us. We ended up estimating that it was approximately 75% complete. We think we had a decent build, but it's not perfect. We ended up being able to predict a whole bunch of orthologs, almost 2,000 orthologs, which we think will be useful for other sorts of evolutionary studies. A whole bunch of SNP markers, which Paula and Anne were very happy about because they can use these for their population level studies. And some of the, a whole bunch of novel stuff. One of the things we did do is we sequenced what are called small RNAs. And they're these little tiny RNAs, anywhere between like 20-something to 100-something base pairs long, and they're used to regulate gene expression, and specifically in what's known as an epigenetic control. So that's something that operates on top of your regular DNA that normally isn't necessarily coded within your DNA. You wouldn't see it there. And we found a whole bunch of novel things as well as some interesting things with embryos. I'll actually go over that in just a little bit so you can see how that works. But what we found is that when we generated our genome sequence, we actually generated about 26,000 gene predictions. And out of those, about a little less than half of them, we were able to identify. Actually, a little bit more than half, excuse me. We were able to identify, and a lot of them we were also able to assign function to. So we were able to actually figure out what this stuff was. So we were kind of pleased with that because that meant that a lot of what we were finding was definitely real stuff. And then we had all this unknown fraction where it could be real, it could not be real. We don't really know. But generally when you do these de novo genome sequences, well, this one may not be the best one. For other ones that I've done where I've had a much more confidence in it, you just end up with a lot of stuff. You don't know what it is, but it's there. It's real. And you have to figure out what to do with it. When we looked at the quality of our genome sort of relative to other genomes, these gray bars indicate when you have a lot of ambiguous sequence. And so if we looked at just these orthologs that we were able to get back, you can see that there was a bunch of these other sequences like Oikopleura, which is a tiny, tiny genome. And they've got a lot of ambiguity in their genome sequence, as well as a lot of codon shifts, which may be true or may just be assembly noise. So we think we did a decent job in assembling this thing. And my image doesn't want to work, but that was just one from the beginning. This is a little thing of the different species of urochordates. I had some problems with this. I'm not sure what's going on. But so here's Salpithompsoni, and here are the other urochordates right down here. The reason why I wanted to show you this is one of the things after we got all these orthologs we decided to look at is if there's any evidence for genes that might be of particular importance for adaptive evolution. And one of the ways we do that is we look at these genes, we say, okay, is this gene evolving faster than we would predict than we see in other species? So we look at a whole bunch of other species and we say, well, is the evolution that we're observing in Salpithompsoni exceptional as compared to all these other urochordates? And so we did an analysis. This is often called Darwinian selection, but it's supposed to reflect sort of directional natural selection. We did an analysis and we looked at the 5 species of tunicates and a species of what's known as a cephalochordate. It's kind of looks a little bit fishy if you haven't seen one before, but it's not. It's an invertebrate. Again, it has sort of early developmental stages and I think a notochord, but no bones or anything like that. And basically, we used that as an outgroup, and we looked at approximately 900 of these protein sequences, of these orthologous groups of proteins. And we find there were about 36 of them that looked to be under positive selection, that looked to be sort of exceptional within Salpithompsoni. Now that could be representative of Salpithompsoni being important, or it could be representative of being important to Salpithompsoni, or they could be representative of being important to Thalassia? We don't really know because we don't have another Thalassian genome sequence or things like that. So in that case, like our inductive approach where we say, yeah, we know everything because we sequenced everything. Well, we don't know all these other species, so we don't know for certain about the other ones. But it is something that then tells us that these are targets that we might want to be interested in following up on and continuing to see what's going on. And approximately 47 of these sites were not associated, that we noticed that were rapidly evolving, were not associated with any protein domain. That means that they're probably novel functional regions just to these Thalassian salps, right? So we don't know if it's Thalassia or salps, but these have novel function that have never basically been described before, as far as we can tell. One of the things that we noticed is there were a few immune genes and ribosomal genes that particularly we noticed being under a lot of positive selection, and that they were also receptive. These genes were receptive. In a separate study, we found that these genes were receptive to changes in environment. So whether or not you were under in a warmer or colder environment or different times of the year, you might see differential expression of these genes. So that got us thinking about, well, what's going on with these? They're under positive selection, and it looks like they're important biologically to the species. I'll come back to that in a second. So when we looked at some of the other things that we found is I mentioned these novel epigenetic factors. Oh, sorry. So if you remember, I got mixed up here. If you remember, one of the things that I said before is that when we looked at ribosomal genes, there were a whole bunch of them in this repetitive fraction, this stuff that we couldn't even assemble, but there was all this ribosomal gene copy that was in this stuff. So it kind of got us thinking about, well, why? So this is making it hard for us to do a good assembly or a better assembly. And there seems to be quite a bit of it. I mean, this is a pretty big part of your genome. We've got a whole bunch of ribosomal genes that look like they're a positive selection, which might indicate that there's lots of copies of them too. What's sort of going on? And so we're trying to think, is there any reason to think that the amount of copies of ribosomal genes in your genome would be adaptive in any way, shape, or form? And so we started looking around, and we found a whole bunch of studies that basically show that if you look at our DNA copy numbers, that they're positively correlated with gene expression, and they're negatively correlated with mitochondrial density. So in other words, the more ribosomal genes you have, the more gene expression you have, but there also seems to be a relationship with mitochondrial density, which is kind of an interesting thing to think about because we do see that in some species that there is increased mitochondrial density in cells to compensate for aerobic capacity. So you're not able to do your aerobic metabolism as fast as you hope to, so you build up a lot of mitochondria in your cell, right? Well, that's often what happens to polar organisms, right? So we ended up with this odd little conundrum here about what's going on, but what it seemed to indicate is something that's really worth looking into is whether or not variation in genetic mechanisms, if this repetitive variation, if this dosage load of rDNA may somehow be related to genetic mechanisms for polar adaptations, right? And there's some evidence to suggest that there are these relationships between ribosomal DNA and responses to different types of thermal stresses. So it was really cool. We did not expect to see that at all. And that's something that we just picked up just by going through it. And it's something now that we're like, well, this is really interesting because we don't see this high level of rDNA in all these other urochordate species, even the ones that have larger genomes. They don't have this same load of ribosomal DNA. But none of them are polar. And so this is definitely something to keep our eyes on as other polar genomes come out. We'll definitely be interested to see if any of this stuff plays out. One of the other cool things about this group too in general is that if we look at basically the evolutionary rates, so how quickly all those orthologs were evolving in general, what we see is they evolve at a rate which is like the same rate if you were to consider the difference between human and birds, right? That's how fast these species are evolving. Like they're evolving really, really rapidly at a protein, at a DNA level within coding sequence. And what we find is that is that, in Oikopleura, which has the smallest one, for a lot of times these smaller genomes were seen as being, well, they must evolve quickly because they're small genomes. But when we sequence some of these larger ones, such as Petrillus and Salp, we notice that that's not the case. It's not that there's something about genome size which is driving this, but there's something about being a urochordate which promotes a rapid rate of evolution. And so it's something curious about the group. We don't really know why that is, whether or not it's a response to natural selection, it's a high rate of mutation, large population size. We don't really know, but it's a really interesting little component that we're interested in trying to follow up on. And so last thing I want to talk about is these epigenetic controls. So this is an area of interest for my collaborator, Rachel O'Neill. This is sort of her specialty. And one of the things that, they've got this sort of complex life cycle. So we did a couple samples of the males and the females, of the adult males and females, and then also we did an embryo, some embryo samples. And basically we take that sample and we mash it all up and we isolate these tiny little RNAs, so just the smallest section of these little RNAs. They're called microRNAs or small RNAs, and then we sequence those. And so what we did that is we found that when you look at mRNA expression, that it's significantly different between these different stages. So if we look at embryo, which is here in the pink, and female, which is here in the yellow, and it's actually just overlapping with male as well. You can see the modes. There's a definitely distinction in the modes. And this is the size of the product right here, right? So this is how big the microRNA is, and this is basically a measure of frequency. And one of the things that we know is that specific sizes, like these 28 base pair size, like these are a specific type of microRNA. And that specific type is what's known as a Peewee interacting RNA or a piRNA. And it seems to be particularly important in these embryos, which is interesting to us. We're like, well, that was an interesting thing to find. If we look even more closely, we see that we even found completely novel, never been described in any other way in any other species, novel microRNAs that are differentially expressed depending upon which tissue size you are. So the expression of these different RNAs seem to be probably really important in the development of this organism. Right? And so we have different ones that are expressed in embryos, different ones that are expressed in male and female. And then one of the cool things that we looked at is a lot of times what these little microRNAs are doing is they're knocking down all these repetitive elements that are in the genome of these urochordates. So all of us our genomes are just full of repetitive elements. It's basically, they're typically the remnants of viruses that have invaded our genome and left copies of themselves. Right? And they're always moving around, making copies. One of the things I work on is placentation, right? And placentation in mammals basically the evolution of that is all attributable to the co-option of retroviral genes that we've done. We've stolen retroviral genes and then we use them to do stuff. And so, but in general, like we're trying to knock down these viruses from doing things. And so you can see that there's this distinct difference in sort of how these peewees are working. This is all just peewee expression, what their targets are in different tissues. So in male and females, they're specifically targeting these DNA transposons, and then we see in embryo that there's a shift, and for some reason they're targeting satellites and simple repeats. And we don't know why that is, but again, it's an interesting novel mechanism that we're trying to figure out what's going on. So there's a whole bunch of different new areas of research that we've been able to, and different hypotheses now that we've been able to generate from all this little inductive study, right? We've got at least 3 major ones. Is our DNA somehow related to adaptation, to thermal adaptation? Is the expression of novel piRNAs somehow involved in this process, and that there's no relationship, that there's this increased rate of evolution that we see within these urochordates that we'd love to be able to figure out why that is and what the mechanisms that underlie it. So that's sort of that one study. The other study that I'm going to talk a little bit about is going to be some work that I've been doing looking at horseshoe crabs. And this was done with a whole bunch of people from various places. But again, this came out of Rachel's interest, and she was interested in horseshoe crabs for a couple different reasons. One of them is that we basically think that they're like a living fossil. They effectively look like they have 400 million years ago, which is pretty wild. So they have this really basic conserved body plan. But they do exhibit patterns of evolution. Like we know that they're locally adapted across their population. But the Atlantic horseshoe crab is just kind of this wild organism. And it's a really important organism from an evolutionary standpoint because it's seen as being at the base of all arthropods. So understanding this species helps us then understand almost all other members of Panarthropoda. So it's a pretty important species from a position-wise from an evolutionary perspective. And then the reason why Rachel was interested in is because she's always been interested in this process by their blood. I don't know if you're familiar with their blood, but they have blue blood. It's copper-based blood. But basically every single parenteral or intravenous solution in the United States has to be tested for sterility using horseshoe crab blood extract, right? So what they do, they're heart puncturing these horseshoe crabs and bleeding them out, and then they let them go, and supposedly they live. They actually do live. Most of them do, I think. Or I don't know in nature, but I've done it to them in captivity, and they've been okay. But it's kind of crazy that you're heart puncturing them and they're all right. But basically they use this LAL test the horseshoe crab blood is incredibly sensitive to any kind of bacterial presence, and it'll form this clotting response with even the tracest amount of contamination. And so that's why we use it in medical sciences or in medical fields to test all this sort of stuff. And sort of the basic premise of how this mechanism works is understood using this response to proclotting and clotting enzyme. But in actuality, there's a whole suite of other things that are going on here. And so we were interested in it, trying to figure out, what else is going on in horseshoe crabs. So what do we know about the genetics of horseshoe crabs? Well, there's about 52 chromosomes, and up until recently, there wasn't much of a genome build, although there recently was just published a genome build not too long. It actually wasn't published, but it's publicly available out of University of Washington, and Washington University. And this sort of lack of information originally got us interested in this. And so we thought, well, it might be fun to do a transcriptome of the horseshoe crab blood, see what genes are really active, and see what's going on in this genome. And so we did it. We ended up with a much better, what we think is a much better genome sequence of this species. And what we found was that there are about 20,000 predicted genes. There were about 13,000 of them that were highly expressed in these amoebocyte blood cells. And we found a whole bunch of things that are related to immune function, and approximately 726 transcripts that specifically were related to immune function that we found there. Of those 726, probably more than 50% of them were things that we weren't able to easily describe. And about out of the total, so not of the 50%, but out of the total 726, almost a third of them we still don't know what they are, but they have novel expression. We know that they have some sort of immune function by some levels of similarity to things, but we really don't know what they're doing functionally. And this goes from everything from antibacterial compounds to JK stat upregulating immune processes. Like there's a whole bunch of different things going on here. One of the cool things that we noticed, though, is that from a more of an evolutionary standpoint, when we started looking at the taxonomic distribution of genes that were similar, we noticed that there was a lot of things that were similar to other arthropods, which we would expect, right? But then we found all these genes which looked like they had nothing to do with arthropods, which was kind of weird to us. Like, well, what's going on there? And then there's a whole bunch of stuff we have no idea what it is. But this was kind of an odd thing for us. We're like, well, this is a really weird distribution. Part of it you could say is just reference bias, because the human genome and the mouse genome are really well done. So when you go to map all your unknown things, like they might map there just because those are the best resources. But even when there were good resources, we found a lot of them mapping to these other groups. And then when we looked at them again, we also noticed that there were specific processes going on. It looked like we had different, so if we looked at the things that mapped to arthropods, they almost always tended to be, they were generally disproportionately related to functions having to do with signaling. So that would indicate that, the signaling processes look like they're evolving sort of at a regular rate or a regular rapidity. While these base level metabolic functions were all disproportionate, significantly so, that's why they're red, significantly more associated, significantly more than random associated with non-arthropod groups. We think that this is actually a signal of a historical signal within their genome. So they basically have maintained some sort of ancestral signal that's significantly divergent away from other arthropods. So arthropods have continued to evolve away from it. Horseshoe crabs have basically stayed the same, and that's left them with genes that are going to be similar to more similar to vertebrate genes than any other arthropod would have. Another really interesting area that we looked at was we looked at Hox genes. I don't know if you guys remember Hox genes. You probably don't. Probably most of you had them in in developmental biology as an undergrad or something like that. Maybe they talked about them there, right? But Hox genes they're really well known, and one of the reasons why people talk so much about them is they're like these key developmental genes that are really well studied. And one of the cool things about them is they always occur in these. They always, we'll put that in quotation, occur in these clustered and collinear groups, right? So they're always inherited as sort of one big chunk. And so they go in a very specific order, these Hox genes, and these are the arthropod version of it. This is a little Drosophila, and the color basically, as they're expressed, they sort of make their way down the organism, which is one of the really cool things about them, right? So like the order that they're expressed goes from head to tail, or head to abdomen in this case. So that's one of the really interesting things, and they help determine, there are all these transcription factors, they're basically determining what the body plan is of this. So if you start manipulating these things and knocking them out, you sort of mess up the entire organism. It can't really function appropriately. It doesn't evolve right. We're really interested in these from an evolutionary perspective, is these clusters tend to be conserved, which is a pretty cool thing when it comes to biology. And when we look across like all different types of organisms, and we look at these they're color-coded for basically the same genes, the same sets of genes, we can see that across all arthropods, we see a very similar pattern. Now, in these basal arthropods, we see less evidence for it, but we still see, some of these labial and proboscipedia genes up here at the top, and then some of these other genes being expressed down towards the bottom. So it's a pretty conserved process, which is pretty cool. If we look at sort of like what the ancestral arthropod looks like, this is an example of, if I took all the arthropod genomes that I could find that were well assembled, or that I thought were decently assembled at the time, and assembled them all and sort of mapped them out to show what they look like, what you can see is that as we look across a bunch of these species, sometimes you'll have sort of these weird inversions. Drosophila has a big break right here that they're well known for, where it's on the same chromosome, but it's on different ends of the chromosome, right? But it's still, if you look across Tribolium, butterflies, water fleas, like all these different things, you'll find that this pattern is still pretty well maintained, especially in the mandibulata. When we move down to the Cheliocera, where horseshoe crabs are, what we tend to find is, first, we have less well-assembled genomes, but secondly, we find that there's a lot more evidence of duplication. So we have duplicates sort of within, like in Ixodes, where we have a duplicative labial right there. We have another duplicate here of sex combs. Like we have these little duplicates, a duplicate of Antennapedia. All these duplicates within this one group. And if we look at horseshoe crab, we get a heck of a lot of duplicates, right? We see that there's at least 2 large clusters right here that have been broken up, right? And those large clusters would indicate that there's been at least one genome duplication event in this species. So if you think about it, this is a species that has not dramatically evolved since for like 450 million years from a phenotypic standpoint. But it's got at least one duplication, probably due to the copy number here, 2 duplications of its genome. If we look largely across, we just look at specific gene trees. And so here we're just looking at we're looking at the labial gene. And every time you see a red indicates that there's evidence, you kind of you can test for speciation events versus duplication events. What we see is basically in this Chelioceret part of the tree, we get a lot of duplication. You'll notice that there's also duplications here in other species. So it's kind of a little bit of an inaccurate assumption that Hox genes are never sort of changing, that they're always fully conserved. There's actually more variation there than what we would expect. But we show extensive evidence for duplication in the labial gene. If we look at another one in Tennipedia, we see another example where it looks like there's more duplication. It does look like there are specific duplication events that can be associated with Limulus polyphemus. But other ones looked at to be more basal within the Cheliocerates. And there's even evidence that perhaps these are basal within the arthropods, which is kind of a crazy thing to say. Now, if we look and build like a giant super tree, can you build this huge tree? And so now we're not just looking at the Hox genes. We're looking at any gene that's got a homeobox domain. And that's Hox genes have a homeobox domain, but this time we're going to look at all of them. So if we look at all of them, basically what we find is we look across this entire tree for all insects and arthropods. We show that Limulus in general, if you take on average, have greater than 2 copies for all their genes. And if you look closely, you can see, for instance, this is labial. Limulus polyphemus is in this dark blue. We can see, it's got 5 copies here, right? And as we go through, so if you look at for these blues, you'll notice there'll be like these clusters of blues throughout this whole entire tree. And so we think actually that horseshoe crab has undergone at least 1 duplication and probably 2 genome duplications, which means that it's got, like, every time you do a genome duplication, you're basically making raw materials for evolution to happen, for adaptation to be able to run wild. Like, that's one of the ways that fishes have thought to diverge so well, because they've got a basal gene duplication. So these guys, I don't know what's going on with them. They've got an enormous amount of genetic material to deal from, to start from. We know that from other studies, from classic studies on them, that they mutate and evolve at the same rate as normal organisms do. But for whatever reason, they are just tied to this phenotype, and we've yet to figure out precisely why that is, but it's something we're really interested in. So there's a bunch of people to thank, especially folks at the University of Connecticut. They were a big help for a lot of this stuff. Of course, all my co-authors, they were co-authors, so I didn't really thank them in here. But a whole bunch of other people who helped with various things. And again, these are just sort of side projects that I work on, and they're kind of fun little things to do, and they do take a little bit of time, but it's really rewarding to sort of be able to do like that exploratory work in biology that you don't always get the chance to do. So that's what I have to say. And if you have any questions, I'd be happy to answer. Or exclamation about something I said. If you don't know. Yeah?
I was wondering if you could sequence a cell closely related, but from tropical locations, do you think that you would similarly see as much repetitive elements in the genome? And I guess you probably think there wouldn't be as much
Yeah.
DNA, right?
That would be one of our hypotheses. Yeah, we'd love to be able to see if that's the case. Or, maybe it's the case that they're not differentially expressed. They're not as receptive to thermal stresses, right? So that's the other thing is like it's not just their presence, but it may be whether or not they're being expressed. Or maybe another one may be do we see patterns of positive evolution in these ribosomal genes as well, right? And are they moving in the same direction as what we see in polar organisms? So, yeah, I think what it did was it basically put us onto this whole idea of like, wow, we actually have to think about ribosomal genes, which, to be honest, we normally would have, I guess it kind of makes sense. They're metabolic genes, but we were, when you're dealing with these large repetitive element sections, you're often just sort of moving away from it, not trying to engage it. And now we're like, maybe now. So doing that would be a great way to test that kind of hypothesis, is trying to figure out whether or not that's the case or not.
And what size of sample would you need to get to get something equivalent to the one you worked on?
Oh, you'd need like one organism. Yeah. So yeah, there's plenty of DNA. And now, like, the DNA, the sample I had dealt with too was not like, it's so much easier to do now. The DNA sequencing technology changes so rapidly that now it'd be like, I don't know, 50, 100 times easier for me to do that project. And to generate the actual data that I need to do a much better sequence. I mean, our sequence is okay. It's not terrible, but it's, I think, our gene predictions are pretty accurate. But yeah, it could be more contiguous. Yeah.
So what do you think, what are the costs and benefits of these genome duplications for the horseshoe crab? They've been around, they haven't really changed, but they're,
I don't know. The only thing I can.
in case they could.
Yeah. And they're euryhaline too, right? So they're like, they deal in all kinds of different environments. So they're locally adapted, and there's no evidence to see that they've lost it. I'm not really sure what the cost is going to be here. I mean, the benefit is, of course, that, the cost for them is probably, it has the opportunity to move them away from a really successful phenotype, and they don't want to do that. We did notice that in their transcriptomes, there's a lot of evidence in the amoebocyte transcriptome. There's a lot of evidence for duplicated genes being expressed and spliced versions of them. Some of them that would be sort of analogous to, a mammalian antibody type of gene, right? So they're either antibacterial or clotting genes, where having more variation there may be helpful in their immune response. So that may be a benefit. It's really just befuddling to me that they've got so much capacity to evolve, and they haven't evolved very much at all. Yeah?
So, going on to what Scott just asked, is the duplication, could it be for adaptations that are really microhabitat level? Could you be adapting to really finite conditions that are really localized spot?
It may confer a plastic response. Maybe that's what it's doing. Again, we only looked at pretty much, I think the sequence was from 2 individuals. So, we weren't looking at within species variation. In terms of the patterns of local adaptation that have been demonstrated before, those are not at very high depth from a genetic standpoint. But yeah, I don't know. I mean, it could be. I'm not precisely sure what it might do. I mean, it could be that responsiveness, since they are a euryhaline species, they deal with a lot of different environmental stresses. Maybe having a lot of a really broad genetic background allows them to be able to persist in very challenging environments that are highly variable. But one of the crazy things about it, they've got, I showed you the Hox genes. Like, the Hox genes are what they need to develop in their, those are like, that's how their morphology is established. And so to have that much duplication in their Hox genes is just kind of crazy. But, they're not the easiest experimental species to work with. We'd love to be able to do a development course with them. And we know what time they spawn and stuff like that, but that was a lot more effort than what we had planned on. So maybe Rachel can convince a grad student to do it.
So can you just describe generally how you go about reconstructing the genome? What's
Yeah.
actually physical? Are you doing this writing script and having a computer doing it, or are you actually looking at?
Well, so yeah, I had to learn all this myself, right? So I started off as a marine ecologist, then I became a population geneticist, and now I'm a genomicist, right? And I will say this, it's to your benefit. It's what I believe, that you should try to pick up some programming as you go through your graduate degree, because it's the way we seem to be moving as a field. And that's what I did. I was at Florida State. I had a guy on my committee, this guy Peter Beerley, who wrote a very well-known population genetics program. And he's on my committee. He's like, you need to make sure you know how to program. My advisor was like, hey, you can take this programming class with Dave Swafford, who's like one of the great evolutionary biology guys. He wrote this program called POP, which was super popular for a long time. So he's like, you should take that class because you're going to use these skills. And they were right. Like, I use them all the time. It lets me do all kinds of stuff. I've used it for environmental chemistry studies too that I've worked on. So there's all kinds of reasons why I would suggest it. But basically, so I had to teach myself how to do a lot of this stuff, because at the time I started it, there was no program in genomics, right? But so what we do is you get your sample. In this case, it would be DNA. So I do transcriptomics, and I do some epigenetic stuff too. And so you just have to get whatever sample you want to sequence, right? You generated a library for that sequence. You submit that sequence to wherever you're going to sequence it. We used to sequence our own stuff, so it was quite a bit more work. We get back that data, and then the first thing you have to do is figure out what the heck you're going to do with all this data. And this happened to like biologists all over the place, I think around like 2010, in that area when suddenly everyone's like, I want genomic data, and then they got it all in. They're like, now I need a postdoc to analyze this genomic data because I don't know what to do. So basically what then happens is you take the data, you clean it up, you'll then run it through some type of assembly pipeline, right? And there's a couple, if you want to do yourself, I would point you towards a couple papers that have these efforts known as the assemblathons, where they do all these comparisons of different assembly approaches, and they give you like a really good, nice test, because it's a test to see who wins, right? And who assembles a genome the best, and they're already like pre-assembled genomes. And so they'll figure that out, and then you can figure out, oh yeah, this one works best with this type of data, this one works with this type of data. So you'll assemble it all, but then you have all this. So great, now you've got assembled DNA sequence. Then what do? Then you have to annotate it. And trying to figure out what is what and the right way to go about doing it is a challenging part. So I'm working on a shark right now. Sharks have very big genomes. And one of the hard things to do is to figure out what all this repetitive stuff is in the genome. Because typically, as biologists, we're often interested in the genes. So we want to figure out what the genes are. But before you can predict all the genes, you kind of have to black out all the repetitive stuff. Otherwise, you end up with a lot of false positives. So you'll predict all these repetitive elements using, there's a couple different tools for that. You'll then predict the genes. Now you've got your genes. Now you've got your repetitive elements. You may have predicted other things like start sites or other stuff like that. If you have multiple samples that you've sequenced, like I sequenced a couple different individuals of the shark species, I can then generate population markers like SNPs and things like that. And that's sort of like the basic process that you do. If you have, you probably, when you do these genome things, sequencing efforts, I would suggest doing a transcriptome as well, sequencing some RNA, because when you do your gene prediction, if you actually know what some of those protein sequences, those coding sequences should look like, it helps the process, because it's like a machine learning training process. So it's good if you have a training set that you can train your data on. But then after you do that, then it's up to you what you do with it, right? And it's interesting because my background, I used to be a wet biologist, and then I was, now I'm by wet, I mean like wet in the water biologist, right? And now I'm more of a wet lab biologist. I still get in the water every now and then. But so from my perspective is like I used to, have to deal with making figures and investigating data. And I understand that that's what you do, but a lot of, like you look at our genome papers, there's like 8 people on the genome paper or something like that. You look at some of these papers, there's like 40 or 50 people. And I've been involved with one of those larger efforts through the Baylor Genome Center. And in that, like no one looks at the detail. So when you're doing inductive work like this, like you kind of have to really dig in and investigate. Like just when you're doing your own research studies, like sometimes it's the details that really matter. And so that's one of these things I think in a lot of these genomes, like that's what they're missing is that, some of this is haphazard when you find it. Like I did this whole work on evolution of complexity in these pieciliate fishes that have placentas. And I was going through it and I noticed, I was like, why are all these viral genes being expressed in these placentas? And I was like, oh yeah, that happens in mammals because they co-opted retrovirus genes. Oh, they've probably done the same thing. That's pretty awesome. That means that's a general mechanism for evolving a placenta, right? That I found in a completely independent lineage, evolving multiple times within that lineage, right? And it was really sort of, I was basically looking at it saying, I don't get why this is there, right? But I was going through that list. And so some of it is actually looking at it pretty closely like that, looking at trees. I go through a ton of trees because I think that that's a useful thing to do. But you can use some of these data mining methods to target the ones that you're interested in. You can program and leverage some of these things. So I don't, when I do my positive selection analyses, I don't go through and run PAML individually on every one. I program it so it runs PAML on all of them, and then I go back through and I look and see which ones I got a positive hit on, right? So That's the general process. But it's a mix of both. I think if you do it well, you still look at the data. It's daunting to do so.
Okay, well, we've hit the 5 o'clock mark. It's, I believe, going to be happy hour for the students tonight, for all of us. And Nate said he'll stick around for a little bit to join you for a little bit more of an informal chat. So anybody interested in computation